I'm John, and I'm here today to talk about an exciting novel study about sexual orientation and serotonin, a neurotransmitter. So in the past, science has been trying to find some sort of gay part of the brain, or in other words, some sort of neural substrate where scientists can find activity or some sort of evidence to suggest a link between that part of the brain and sexual orientation. So for ages, people have been asking, are men born gay? Male orientation is likely a factor of biology and social factors, but mostly biology. Things such as genetics, epigenetics, which are genetic factors of the mother or the parents that influence the child, the prenatal environment, such as different sex hormones that are going around in the womb, and the postnatal environment. In other words, how does the brain develop as a result of different hormones and other environmental factors very shortly after birth? So one famous study that happened in 1991 by Simon LeVay, the author of our textbook, was a study he conducted on the postmortem brains of homosexual men, heterosexual men, and heterosexual women. So Simon LeVay had been reading a number of studies on rats and many other animals that had linked sexual preference to a specific area in the hypothalamus, which is in the very middle of the brain, called the medial preoptic area. So Simon LeVay wanted to see if there would be any sort of correlation between sexual orientation in men and this very specific area of the brain that had been found to control male sexuality in a number of different species. So what he did was he selected 41 brains from people who had just died. 18 of those were homosexual men who had recently died of AIDS. 6 of those were heterosexual women who had died of different causes, and 16 of those were heterosexual men, some of which had died of AIDS. So he dissected the brains to look at different sexually dimorphic nuclei in the hypothalamus of these different individuals. And he found that in one specific area, INAH3, that there was a significant difference in the size between homosexual men and homosexual men and heterosexual women and heterosexual men. In fact, heterosexual men had an INAH3 that was twice the size of homosexual men and women and heterosexual women. However, there were a few weaknesses of Simon LeVay's study. Namely, the study was only observational, and it occurred at the time of the HIV/AIDS crisis. So all of the men who were in the homosexual group had died of AIDS, which could certainly be a confounding factor, considering only some of the men in the heterosexual group had died of AIDS, and none of the women had died of AIDS. Additionally, there was no way to determine whether the heterosexual individuals in the survey were at all homosexual to some degree. So this certainly limits the effect size of the study. And again, it's only observational, it's not experimental. The sample size was also relatively small. There are only 6 heterosexual women in the study. And a replication of the study found slightly different results when it came to heterosexual women. In fact, we found that homosexual men had an INAH3 that was sort of in between the size of women and heterosexual men. And of course, there are a number of limitations. It's of course a post-mortem study, so it's really helpful for determining the anatomy of sexual orientation, but it doesn't really tell us a whole lot about how it happens. Rats. So now I'm going to focus on the next portion of my presentation, which is rat and animal studies. Why would we study rats and other model organisms? So we don't encounter the ethical and legal implications of studying on humans and cutting them open and dissecting their brains and all the other gross things that we do in animal studies. Further, but it's not just the issue of cutting people up. It's a lot more than that. There's a short reproduction time with rats, and the life cycle allows for developmental studies that we wouldn't be able to realistically do in humans. Additionally, there's a lot of existing science around rats and their biology. So we have a much better working model of the brain and allows us to make much more fine-tuned advanced experiments to come up with findings that we wouldn't be able to come up with in other species. So what do we know about rats and male sexual preference? So it is very widely established that the area I previously talked about, the medial preoptic area, is associated with male sexual preference, motivation, drive, arousal. Many studies have found different effects when things are messed with in this area. For instance, lesions in the MPOA have resulted in changes of sexual preference between males, females, or no preference at all. Development of this area is determined by sex hormones and other chemicals in the brain. And findings about the significance of this area in relation to other animals has almost universally yielded the same result, that it is the controlling area of male sexual motivation, which shows that this area is, evolutionary speaking, is really old and can probably apply to a lot of species, including humans. So what does this all tell us? It tells us that we can apply some of these studies when it comes to replicating in humans. So a study we might read about rats and serotonin or any other area might have implications for humans. So let's talk about the physiology of this area. So we don't know, again, this really emphasizes the importance of physiology. We don't know where sexual orientation, we know where sexual orientation is in the brain, but we don't know how it really happens. The two studies I mentioned only show that that area was associated with sexual orientation. It didn't tell us how it mattered. So one possible direction to explore, which I'm now going to talk about, are networks of neurotransmitters. The neurotransmitter I'm talking about today is serotonin. It's found throughout the nervous system, particularly in the gut and the brain. And low serotonin is usually associated with psychiatric disorders such as depression and anxiety. But serotonin is not just about depression and anxiety. It's not just used as a medication for antidepressants. Serotonin has a huge influence on sexuality. Levels of serotonin are inversely related to sexual arousal. So medications that increase the availability of serotonin in the brain often lead to sexual side effects such as low sex drive, erectile dysfunction in both men and women. Going back to the medial preoptic area, numerous studies have found that serotonin has a huge role in this particular area of the brain. And experimental studies have shown that altering the levels of serotonin, either during development or even just acutely, can make huge changes in the sexual preferences of animals, as well as their motivation and behavior. And further studies have shown that administering drugs that alter levels of serotonin can also affect behavior or modify or change pathways in the brain in different ways depending on sex or sexual orientation, which shows a sexual dimorphism. So why would we not potentially investigate these effects in humans? Or why hasn't really not been investigated at all? Well, there are a few problems. It's really hard to extract people's brain chemistry. It's obviously unethical to kill people and dissect their brains, and the current technology is very limited and only allows us to determine so much. The medial preoptic cortex is a very small area in the brain, which makes it very hard to sort of analyze in vivo, or while an organism is living. And this brings me to my focal study, Kinnunen et al. 2004. So, Kinnunen et al. Was a group at the University of Chicago that wanted to investigate the role of serotonin by administering fluoxetine, which is a selective serotonin reuptake inhibitor. Selective serotonin reuptake inhibitors block the absorption of serotonin by neurons in the brain, so more of it floats around freely in between the neurons. The study was counterbalanced and double-blind, which essentially means that the participants nor the study authors knew who got placebo or fluoxetine. So there were 16 participants in the study. 8 of them were exclusively heterosexual and 8 of them were exclusively homosexual in order to maximize the effect size. Each individual was scanned under both placebo and fluoxetine, which makes a counterbalance with a week-long gap to mitigate the effects of the administration of fluoxetine. So the study authors used a PET scanner to measure the glucose metabolism of the study participants after the administration of placebo or 40 milligrams of fluoxetine during a standard visual monitoring task. And they found some pretty striking results. Over here, you can see the PET scans of the heterosexual participants, and here are the homosexual participants. Red corresponds with an increase in metabolism, while blue corresponds with a decrease. And as you can see, there is a huge difference in how the homosexual group responded compared to the heterosexual group. So what can we make of this? Even though this is a very early study, in fact, it's the only study to show neurochemical differences according to sexual orientation in humans. Since it's the only study, we can't say a whole lot, but it really shows us that there is a lot of direction in science that could be forthcoming around the science of sexual orientation. It shows us that as technology advances, we can find a whole lot more about why sexual orientation develops and happens the way it does. It also shows us that this area has a lot of analogs with rats, not just in anatomy, but even physiology, which really paves the way for further studies in terms of the evolution of sexual orientation, and further proves that sexual orientation is a highly biological